All right. So, I guess I should say something about myself. I'm Australian. Uh, so I finished my PhD not that long ago in South Australia. I've mostly worked on Homo erectus and other like phylogenetics, biogeography, evolution, that kind of stuff. But now that I've moved to the US, albeit briefly, I was working on, I guess, bee conservation and macroecology. And I guess the first step for that was to get the data together and get it cleaned. So I guess that's the context behind what I'm about to show you. I don't know exactly how polished it'll be, but I think maybe the best way to all of the available public data. And I'm assuming that some of you have tried to use these data before and come across some of the common problems such as poor names, this, that, the other. So the idea, I guess, was to get rid of those problems and publish something that hopefully everyone will be able to use. So we don't need to all wheel every single time we want to publish. And hopefully just stimulate a whole bunch more research. So I'll share my screen. And I'll start off just my. Everyone see that well enough?
No, I can't see your screen. And if you do freeze up,
yes.
I'll just type something in the chat. I think you would read it. I'll just type frozen or something. You froze up for about 10 seconds.
Yeah.
And anyway, go for it. This looks great.
Yeah, maybe I should have stayed where I was. My internet has been kind of unstable here. Anyway, hopefully that won't be a problem. So basically, I've tried to get together the data major repositories. So that includes ALA, Scan, IDIGBio, and GBIF. And I also have the USGS data from Sam Drogi. Thankfully, ALA has really good R package, so I can actually download the ALA data easily for all within R. And I've tried to make a whole bunch of functions, which should just make this really simple and a few lines of code, and they always hide. It's all of this. So it takes up 200 lines of code. So instead of having that, we just have the one command where you put in the folder that you're working from, your email, and the taxon you want to download. So in this case, I've gone with the Apiformes, which is just all bees. Then, because Scan, IDIGBio, and GBIF, some of them have R packages, but none of them are particularly good. You're often limited in how many you can download. I found that the easiest way to do this was just to, and I'll share my whole screen now actually, was basically just to put all of these downloads into one folder. So you can see here, this is the path made from that first function. But then we've got, the GBIF data download, Scan download, IDIGBio download, and USGS data. And to try and make it as simple as possible, all you need to do is have them all in one common folder, and this function will go through and find each data type, import them and merge them, and then save them as either an R file or a CSV, or a bunch of CSV files. Once they're saved, if you want to come back to it, you can just import it as well using this. And I've also made, for the time being at least, a USGS formatter. So it just takes in the USGS data and formats it to like a Darwin Core standard that can be fed into the rest. Hopefully in the end, I'll make it possible for people to import their own data sets this way. So I might make a generic format of this. So people can have all of the online data as well as add in their own if they're working on a specific area particularly.
Can I ask you a question?
Yeah, of course. Feel free to ask throughout.
So you're downloading data from the front end, not using an API for a GBIF, iDig Bio and Scan?
Yeah, that's right. I was trying to use their R packages, but for example, you probably know GBIF has a limit of, I forget how much, not very much.
200,000, right?
Yeah, that's not going to get you very far. So I tried to write like a function to just download by species and just loop through, but it never matched how many you download from the front end.
Yeah.
And it was a lot longer. Yeah, so that's the only annoying thing, but it should be easy enough anyway, because the importer will actually find and import all the metadata as well. So it stays with the file automatically.
So I'm sorry. So James, so this, what you have written, it basically just automates a process where it goes out to each of these providers and sucks all the Apiformes data.
No, because you need to download it first. Only for ALA was I able to make a wrapper function that actually got it directly from the API. The rest you go to the website and download. But it'll import and keep as much of the information as is provided in terms of the search terms you use, DOI, dates, that kind of thing.
Okay. Well, now I'm a little bit confused. I mean, so first you just go to the website itself and download the data?
Yeah.
Oh, okay.
Yeah. Because none of them but ALA have a R package or API that's kind of fit for purpose, I suppose. And I could continue to try and make it do that, but it would end up being like you'd leave it running overnight, probably for something like GBIF, because it would just go through and download by species or by genus and have to merge them all together. Yeah. And there's more room for failure there, especially using loops. There might be a nice way to do it, but none that I can think of easily. This is just the easiest. And you'll keep all of the metadata with it still.
Does GBIF have a limit if you download from the front end?
Yes. Oh, sorry, from the front end? No, I'm not sure. I haven't hit from the front end, I haven't hit a limit yet.
Yeah, okay.
Yeah. ALA does for the R package as well, but it's 5 million. So we're not even getting close. It has 200 something thousand.
So they obviously they have it set up so that you don't drain their system. So there could be a lot of people sucking 5 million records.
I don't know. They've never complained to me, and especially when I was testing it, I think I ran it like 6 times in a couple of hours or something. I almost wondered if I was going to get a snarky email from ALA, but it never happened. So presumably it's not a problem, but I don't know what infrastructure they have.
Okay, cool.
Yeah.
Thanks.
Yeah, no worries. Yeah, and so anyway, each one of these functions also has a bunch of helper functions. But in the very end, there's this formatted combiner, which will basically combine like the USGS data or any other generic data format that you put in and combine it all together with the rest. So you should end up with a bigger data set, but I'd like to be able to get, I guess, other data sets eventually to test with and set up a way that's easy for people to input. And it might just be, if you've got your own data format, you might put it in and say, lat actually equals decimal latitude or whatever. And then you can just fill out that for your function and it should read it all in. But that's to come once I find and mess around in people's data sets. And then there's just a generic data saver, which should save the entire thing. And it'll also save individually the data set itself. The EML files, which are ecological metadata language files, which are kind of horrible and dated, and I don't actually know how useful they are anymore, but they have a whole bunch of information about citations and the rest of the data, which I can kind of show you later. And then a file prefix, which is just simply fin for me because I said finished. So once you've done all of that, you'll end up with some occurrence file saved on your computer. And then we've got, this is basically, so that's just merging all the data together and saving it. Then we have, basically this is the cleaning workflow, and you can import the occurrences here, which I did yesterday, so I'm not going to run because it takes a little while, but each one of these functions should also tell you exactly what it's doing. So for example, I said import the occurrences. It tells you that it's found dates in the file names, and it'll take the most recent file from the file name. This is the file that it found, and it's an RDS format, so it'll load that file. It tells you that there was no unique identifier column, so it's made that. So basically each record should have a unique identifier now. It tells you that the data set has 12.6 million observations from 59,000 species, which goes down once you harmonize it. And the species have between 1 and 316,000 observations per taxa. So I'm guessing, like I said yesterday, I'm guessing this is the European honeybee with that many records.
How are you dealing with duplicate data from the multiple sources?
Yeah, that's something that we're getting to. So there is a no, it's okay. It's a good question. Why don't we just jump to it anyway? If it's in here.
By the way, James, I bought my slingshot the other day.
Oh, good. For canopy stuff.
Yeah.
Yeah, no, good. So okay, we don't have it in here, but the function which we're planning to build, well, it's built, but it needs to be fixed up. We'll go through and look at the coordinates, the date, the collector, as well as it'll try and match codes, so collection codes or museum codes, to see if they're, and if all of those match, or if some of them are NA. Well, yeah, basically if all of them match, then for time and place especially, they'll be listed as a duplicate, which is imperfect, but it depends, I guess, on people's data standards and data keeping and whether or not they upload things like individual collection code or museum code.
So if a record, if two records have the same location and the same date, even if they were collected by different people, you're counting them as a duplicate? Or
if they're collected by different people, it wouldn't be a duplicate.
Okay.
And if it had different collection codes or different like museum code, it wouldn't be a duplicate. I thought about using like the identifiers for things like GBIF and ALA, but of course that won't work because if it was uploaded once here and once there, then they'll probably be given a unique identifier in each place. So I think it's the best that we can do so far, at least in terms of being, I think it's better to be more conservative maybe.
Yeah, I'm just curious because Paige and I went through this exercise with the data set that we are getting ready to publish, and we went back and forth on what was a duplicate, how are we going to classify this, what's the correct way to do this. So
did you figure out a good way to do it?
What?
Did you figure out a good way to do it, or what were your parameters?
No, we basically did what you did.
Yeah, cool. Yeah, I don't know what else you can do if the data aren't there. Like you can't do anything about it. And I know the way that I collect, at least, I'll go out to one site and collect, 100, 200 bees, and they'll all have the same location, time, and everything, unless they're split into vials or have a museum number. But that involves processing. But once they've been identified, I mean, they should be processed as well. So
yeah, I mean, it would be rare probably, but if you had two people who didn't document their species correctly, and then you ended up having a similar record, but there was information missing, you could inadvertently delete a point that was not necessarily a duplicate. Does that make sense?
Yeah, no, definitely. And of course, if they're different species, they're not going to be deleted. You'd hope that But I mean, of course, like so many of the records also just don't have date or time. And that sucks.
That was something we were finding too.
Yeah. Yeah. Me and some of my other PhD friends are trying to write a paper about people collecting the correct data. Yeah, but I don't know. That might be a losing battle. Anyway, did you guys have a question with videos on, or you just joining
No.
the video show?
You're doing good.
Sweet. All right. So anyway, there's some of this is reading in certain data, or these columns are used internally, and I'm just telling it that these are the column names to give those things. It's also finding a file which has information about flags, because the way we're actually filtering isn't by filtering out directly, it's by flagging all of the records, and giving the user the chance to filter a complete data set. I'd like to have that data set ideally hosted somewhere. And I was mentioning to Neil yesterday about maybe putting it on Scan, because, you know, if you've got 12.5 million records, I don't know that you couldn't deal with that in Excel. It's hard to deal with it in R. And if you could just choose the filters you wanted from something like Scan and then download a cut down data set, that would be nice. And then this is just some information which is talking about this data set specifically. So it's looking at bees globally. I made it with another guy, Matt Rogan, in the lab. The download date, you guess, it's just going to guess today's date, I think. And the spatial reference type. But anyway, so once you've imported this data set, okay, cool, it's by itself. It's actually split. It also has the EML file attached. This is going to look really horrible, so I'm sorry in advance. But this is basically a concatenation of the EML data sets from GBIF, ALA, and whatever else. Some of them have that automatically, and for some of them, I had to kind of just hack together what I could from whatever those download sites have. But, you know, it basically breaks down a lot of the metadata. So you could go in and look at, let's do something small. So the GBIF records for Senatridae, and then it's got all these things. So additional metadata, the data set, yada, yada, yada. Let's just have a quick look and see how ugly it is. Okay, there's not too much there. So it's from GBIF. It was downloaded on this date. There's a citation, the URL to download, alternate identifier. Basically, there's a whole bunch of information so that if anyone ever wants to use the data set again, they know exactly where to find it, and also what search keys and stuff were used to find it. From that point, we basically just go through a whole bunch of steps which flag different things that you might want to filter for. Down the track, I want to turn all of this into one line of code where you just put in, I want to flag missing columns or invalid coordinates or a GBIF issue or imprecise coordinates or just all, and let it run. I ran a couple of them yesterday, so I still have that in my computer, but I'll just run another one now. So let's just do something that's simple. Okay, we can just flag the basis of record. So most of the scripts should tell you what it's doing as it's going. And because there's 12.5 million records, it can take a little while. Okay, there we go. So it flagged 3.4 million occurrences from 5,700 names. And because I've also got a license flag, which flags a whole bunch, because basically it'll flag anything that has a license attribute to it. So there's 13% remaining on 13% of the records remain unflagged. And that information just gets appended to this, which is a bit of metadata. So if I view the cleaning steps, this is metadata that's associated with that file, basically. So, I've looked for whether or not they're missing coordinates, and then here were the Creative Commons ones. There's a short description, the step that it was, flag included because we ran it true, what the column is called, how many points are flagged, how many species are flags, and the points remaining. And then also the parameters of the search. So for example, this one is basically if the lat or long were not there, then they were flagged. And for the rights one, so for anything in the rights license or access rights column, for, in this case, unrestricted use, anything that matched 1.00, CC0, whatever else is flagged, basically. So it tells you the exact parameters of the flag. So it's all obvious to any user or downstream user what you've done.
So when things are flagged, you're saying that there are issues with the record.
It depends. So it depends if you think that they're an issue. So for example, the unrestricted use, that's not an issue, right? Like it's basically saying anyone can use those data. But for the restricted flags, there were 745 points, and I'll go to that one actually. So restricted strings here. So basically anything that had all rights reserved, no derivatives, or not for public. And I was saying, yeah, I put this comment here. I'm like, I'm pretty sure this has been uploaded in error. Like these data shouldn't be on GBIF basically. But someone's uploaded them. So anyway, it's nice to know. And then there's also ones that are only for non-commercial use. There's quite a lot of those. And then it's up to the user in the end if they want to filter those out. So
I just had a quick question. You showed us the metadata of the flags, like the combined number of records, but then you also appended at the end of the occurrence table, like which individuals are flagged.
Yeah.
How do you deal with like a occurrence having multiple flags? That like does each flag gets like appended or?
Yeah. So each flag has its own cohort, like its own column. So the one we just added there was poor basis of record, but there's no coordinate, unrestricted flags, yada yada yada. And it's just a Boolean true or false. So I could do just make a quick table, and then for poor basis of record, that many were flagged and that many weren't. So I was just going to append each record. We'll need to add the duplicates function. I thought it was here, but maybe we've taken it out for the moment. Anyway, that's just flagging. I've also horribly named this btaxo harmonizer, which is another function which should go through and harmonize all the species names. I'm going to be brave and click run because I don't think it takes that long, but I could be wrong. Oh, okay. Maybe I haven't. Oh, okay. I know what this is. Sometimes I'm lazy and I just try and make it require all of the packages in one go. And then it says that object's not found, but it doesn't do it all the time.
I hate dplyr. It's notorious for that.
But if you have questions in the meantime, feel free to
So your harmonizer package, or not package, function, it will parse out like genus and subgenus, ones that have subspecies, that kind of stuff, or is it actually looking for synonyms? How'd you do all of that?
Yes, so it's using two lists. So I've told it through and find John Ash's synonym list. And also there's an extra like misspelling list from the Orr et al. Paper. So these are just saved in the common directory. So it'll use those to correct the names to whatever OR chose. And I don't remember off the top of my head if he uses subspecies or not.
So, Paige, maybe you can jump in here too, because when we started doing this, we found a whole lot of differences, especially the OR list. And maybe you've already like figured these out, but
I mean, discrepancies between the lists.
yeah, remember how, now I don't remember exactly what was wrong with it, but remember how Michael's list was not.
There were a lot of different situations. Like when I I have a lot of questions, but like when I first ran against synonyms, sometimes there would be duplicate pointings. And so it would say this one is actually one of two. And so then what I had to do was, I don't know, kind of like output that file and email it to John and say, Discover Life gave me two options. Which one should it be? And then he told me, and then I had to like manually kind of adjust it. And then, but then the other question I was going to ask is, I have a mixture of code that like removes numbers and removes author and date and all of that stuff, because as you know, that's usually in the cell and it makes it impossible to combine. But if there were an instance where it said like genus, species, and then morpho 1, like it would actually put in there like Bombus huntii morpho 1. And so then if I would to just like line it up against another list and say drop the morpho 1 and just give me Bombus huntii, that wouldn't be accurate for our paper because we wanted to drop all morphos. So I'm just wondering, like little things like that. Did you just
And I just tracked her and she's out in the field behind follow.
Neil, your
you're not
mic is
muted.
Oh, sorry.
But yeah, I think that's by far all the little characters, and even like an extra space when there's like 2 spaces between the genus and species, then that wouldn't get recognized when it was cross-checking against a list. Did you run into this kind of stuff?
So the first thing that I did, which I'm trying to open now, oh damn, different. But I basically made my own synonym list, and I used a really horrible script to basically. I feel frozen. Am I frozen?
A little, yeah.
Okay, cool. All right, now I'm back. Open that up. So, where did I put it? Data.
Does it help if we
That's the one.
turn off our videos? I mean, for your free
I
time?
don't know.
Okay.
I think it should be fine. I think it's my internet being weird. I don't know why. It's only been happening for the past couple of days.
I think it's just easier than just like speaking to black boxes, but
Yeah, it is easier. I'll tell you what, I'll put it onto my phone. So I'll cut out for a second. Okay, can you hear me now? Good. I'm on hotspot, so hopefully that'll be better. All right. So this horrible file is what I've made of John's data set. So I think I probably had a very similar experience to you with John's data set, where it was like split up by text, right?
Wait, did you get this from Discover Life?
Yeah, so I got this from Discover Life, and I basically made a script to fix
Got it. Okay, so we had Nick do that for us, Lindsay, and then
no, I remember that. And but I thought that there were other issues. Like I thought there were issues within this data set and the OR checklist, or not checklist, the OR data set that were contradictory to each other and different things. Because I mean, we went through this, of course, it was just for the North America data, but I mean, it took Paige so many hours to
Well, yeah, and what I can relate to is the fact that the code that the difference between you and me, and the difference between me and like a real programmer, so you, is that like you put everything in functions, and like if I don't want anyone to ever see my cleaning code, but it's so like that's, you and I should actually talk if you're like ever willing to kind of give a mini lesson on just like creating functions.
Yeah.
But I get what you're saying about like, it wouldn't be like, even if you wanted to share your cleaning code with someone else, it's not very self-explanatory. And there's a lot of like stuff that just has to be done in order to get to the point that you're at now. And
right.
That's what took the most amount of time. That took months sometimes.
Right. And the problem is as well that we always redo this over and over again. Maybe I can try, because I mean, like there's so many issues, and it's unsurprising because it's such a big data set. I'm trying to find another script now. One that I haven't, hold on, shared. I put it somewhere stupid. There it is. So I actually made a script to get the data directly from Discover Life.
Okay.
I also have the file from John, but it's basically in the same format. Let me see if I can open up. So this is going to be really horrible to look at. And like some of your code, I try and always make code so that someone can pick it up and use it. But I've made a function that's called name splitter, not very creative.
Mm-hmm.
And it's built based around basically John's data set.
Okay.
And so it'll basically find, so these are the patterns that go in to find the author name. So if it matches any of these, it'll cut from there.
Yeah, I have something similar where then it takes out the author part and you're just left with the genus species.
Yeah.
Uh-huh.
And of course, I don't know, there's the general rule, which is easy, an author name is in brackets with any lowercase or uppercase letters followed by something with a hyphen and then an underscore. That's probably a very specific case, actually. Some of them will have letters, numbers, and some of them I just take from like the second uppercase. It's a lot of shit. Find any words before and. Find a space before the uppercase letter, i.e. after an author name, after the species name. But I've tried to make it so people can take it and read it. And it looks really ugly in this format, but it's less terrible. No, it's still terrible. Anyway, it goes in like this. So it's one string with this, and the bar is an or, this, and then or. This is actually just a test because of all like the mistakes and like little inconsistencies that are like in the thing. And then I just run a function to do it. And I think it takes like 10 minutes to run. And maybe, I don't know. But this is something that we could go through in more detail if you wanted as well later on.
Yeah.
Because it's complicated, I guess. It tests, for example.
Willing to share this?
Yeah, sure, definitely. And especially, well, I don't know what you guys' plan are with your data paper, but like for me, working. Yeah, what are your goals? I think it sounds like from Neil what we're doing is different. But whatever I do with this, I'm very happy to like involve everyone, and I think you guys have a lot to contribute.
Yeah, so we are we started out doing, Paige and I and Neil started out with this paper just trying to identify gaps in the US where bees weren't. And we quickly realized that it was not going to be as simple as just running this easy script. And so what I initially thought would be a like couple month project turned into a 2.5 year project.
Well, hey, for the record, I thought it would be a 1 month project.
But it's in its final stages now.
Yeah.
And we all discussed making the data set, publicly available, and hopefully many more projects will come from that. But it is restricted only to the US.
Yeah, cool. No, great. And my goal, well, for this, I don't know how much you've heard about my time at the Yates lab. It was hard to talk about what we'd do with this eventually, for many reasons. But at this point, I'm split between trying to do this and answer a question with it, or just do this and release all the script, all the data, so that anyone can take it and ask their own questions. And I'm leaning towards that. Yeah, I have questions down the track that I'd like to answer with data like this, but at the moment, I think I've got more pressing things to answer. So the other thing is if someone had a question, but the thing is it's a global question with global data, and I don't know that we have the data to actually answer global questions much beyond that OR paper. I was throwing around ideas yesterday about looking at even just highland species versus lowland and latitude and this, that, the other, and where species are discovered. But I don't know. If there's an interesting enough question that we can actually answer, I'm happy to do it. Otherwise, I wouldn't mind just publishing a data paper and then people can use it and save time instead of just spending as long as you have cleaning data.
This is kind of an off the cuff idea. There was, I don't know if you heard about the global soil data set that was released maybe, I don't know, like a couple months ago. What?
That sounds fun. I didn't know.
Yeah, and so if we had a global bee data set, it would be cool to match up the soil with the bees because, most bees are ground nesting, and you could see that if there was an impact there. Anyways, I did something similar with trees and Megachilidae.
Cool.
And soil would be also really fun.
Yeah, I'm totally down to answer something with it. So it's really like I was hoping to be guided as well by like what people want and whatever is useful. But it sounds like you guys have done something very similar to the US in terms of the data cleaning steps. I don't know where you see the differences.
The differences that I see are, well, I mean, maybe you could say this isn't necessarily true, but for everything we have done up until this point, yes, some of it is reproducible, but there is a small amount that it did require like actual emails from John telling me what to do, and then I like manually type it into a CSV and then have to change that CSV and like that type of stuff. It's not on a Discover Life list right now. So like if 3 years from now or 1 year from now, there is a whole bunch more bee data for the US and someone wanted to add to our data set, it's possible, and they could 100% use all of our range code and our expected richness code. And that's like a lot of the stuff that we're putting up on Git. But in terms of the cleaning code, I struggle with how would they take what I did, which is kind of like patchwork, and then take this new data and mesh it together. And it almost seems like you are like one step further, which is great in that you have these functions. So like the harmonizer function that you wrote, would someone be able to take a new like a set of 50,000 more records and run that harmonizer function on it and take care of all the issues?
Yeah.
Okay. So then sorry.
No, go on.
I was just going to say, so like a large portion of our code can do that. It is automated like that. But what about the like 3 or 4 records that have a unique situation that weren't ever seen before? So you never wrote code to account for that. That's like, that's seriously something I've come across. And I don't know. I just feel like there's always something. And I'm not trying to be pessimistic.
No. It's true. There's always something.
Yeah.
Talking in terms of the names or the records?
Oh, usually the names.
Okay,
my biggest stumbling block was cleaning the scientific name column.
Yes. So yeah, I guess I've shown you like the name splitter thing, right? This horrible script basically is just splitting the names, cleaning it up. It's quite horrible. Like if there's a single name, if there's 2 names, more than 2 names, this is in terms of authorship. There's different functions and there's tests that go into that.
Yeah.
But you get down to once you save the data and then you read it back in, you find the most recent file, read it back in, and then you need to clean it by adding high names, managing flags. So like, the homonym 1, 2, 3, 4, sick, all like the random flags that John has in there. I've actually got 2 columns now, like one with the name without and one with the name with all those flags. And then the flag is identified also in a separate column, depending on what you want to do with the data and what data set you're working with. And then you can match across all the data sets. But I've also got add tax on rank, add valid, manual corrections.
Yeah.
And so let's see, I should actually, hold on, set my home path. Manual corrections. Oh, damn it.
Well, while you're doing that, so is your hope to publish not just the occurrence data, but also the new synonym list that could be read through these functions?
I would need to ask John.
Okay.
Is what I would say. Because, for example, this script as well, I'm not gonna share the first part of the script with anyone without asking John. Because it can scrape the data from Discover Life. My idea is then I can just get the most up-to-date version whenever without having to bother John. But it's not the kind of thing that I'm wanting to give out to everyone.
Yeah.
For John's sake. But I doubt that'll be a problem for you. But let's have a look. So this is my manual corrections CSV, and the idea was behind this that anyone can take it and add to it, right? So I will give it to people, and if they find new things, they just append this. So this actually has all of the columns. This is basically just the columns that you want in the synonym list. And at the very end, it's got match to valid names. This is like the valid name that it will match to. And the notes, the problem with this one was that there was a var as a species name. So because there was no subspecies, and then it was Bombus var cainensis, it picked var as a subspecies, and it probably put cainensis as like the subspecies, and then it may have even been subspecies cainensis drainardi. Anyway, it's just an unusual case. And then there's other ones, species as SP as a species, and then the letter as subspecies. So the species was sp and the subspecies was a. Missing genus bracket.
He's saying that the letter is a subspecies.
Well, that's what the script does.
Oh,
so because errors.
Yeah, these are errors.
Got it.
And these are ones I'm like, you know what, it's probably just easier to change it manually, but I don't want people to have to change things manually because, as you say, like it's not necessarily reproducible. But this way you can just fill out this, share it with your publication, people can add to it, they can see what you've done, and then you've just got the notes for what the problem is.
Mhm.
And then you can see this is one thing that I want to email John about. So is this a mistake?
Right.
Like the author's back? I don't know. No data, just authorship. Like that's a weird one. Yeah.
I would say that what's like, I have something similar, but instead of having all of these columns, what I have is like the scientific name from the download, and then what it got changed to manually by me, and then a bunch of reasons why, kind of. So like it's a similar thing where you have to actually create this CSV manually, but we are sharing it with our with our publication so that people, I suppose, could add to it, but I don't have all of the all the columns that you do.
Well, it's not a problem. It just means that I've gone through and manually changed what's in the data, and it'll just match it back. And in fact, if I were to use, I'll eventually turn probably like a lot of this into a single line of code. I'll just make this into a function. So you'll probably end up just putting in like the path to your folder, maybe the names of the files that you want to find, but otherwise I'll probably just make it so I can find them automatically, and you can add it in if they're different than what's expected. So then you'll end up with something like this workflow and not this, like running line by line. The problem with making functions is you need to have like lots of prints. You know, there's a problem here or no, what the problem is, how to fix it. You need to anticipate how people are going to break it. Yeah. So I don't know.
And so like for somebody to run this on like a different set of species, what they would need is like download the data from like the front end from like gbif and all those things, and then have a synonym list, and then they could run through this pipeline.
Yes.
Okay.
Hopefully, yeah. It's always interesting until you run it on a different group.
Yeah.
I don't know what's going to happen, but I think, you know, I'm pretty sure in the end, I turned my synonym list into something really basic, which is basically like, you know, this is the accepted name. Here is a synonym. If you find this synonym, make it this accepted name.
Yeah.
So like in that horrible big synonym list folder has a whole bunch more information, which you don't necessarily need.
And I noticed like some of your flags were like imprecise coordinates and things like that, but you haven't gotten around like cleaning that. It just flags it and tells you like these ones are imprecise.
Yeah. I mean, it depends on your question, right? I mean, like if
Right.
it's imprecise, but you want to go to the country level,
Yeah,
state level, maybe that's okay. Maybe it's
and
not.
like, are you cleaning things like date as well? Like,
Yeah, we've got them in as flags.
Okay.
I guess it again depends on your question. I mean, like coming from Australia, so many of our records are from the 70s.
right.
So like if you had to clear from the 70s, you'd get sweet FA. And a whole bunch more at NA. So I don't know. It really depends on your purpose. And that's the thing. Like I don't want to anticipate what people want to use data for. I just want to provide it to them at this point.
But I mean, just like highlighting the flags and like fixing the names, like that's like a big step. It's a long way there.
Definitely. And I've you know, I guess the main paper that I've done, which uses bigger data, like I didn't even think to harmonize names. And I know that there's like homonyms, 2 homonyms in there because I ignored subgenus as well, which I shouldn't have. So I don't know. It's just hard to know. Like it's a learning process for everyone, but if it's just straight up there, great.
Right. Well, so in terms of use, and we lost Angela and Lindsay.
Well, Lindsay had to go to another meeting at 2.
Oh, okay. So we lost Angela, but Oscar's here. And so right now, Oscar and Angela are working out, seeing whether the ellipsoid SDM method is at least as good as the Maxent method. And then we are going to, it's sort of undefined, but so we're going to go at least to the North American level, and how far south we go, I don't know, to develop one or more ecological niche model based papers. And so, and so if it wasn't clear, you're invited to participate in this paper. So, and so Angela's, she agreed to lead it, and now Oscar and Angela are leading it. And I just hope by the end it's not me leading it. But
who knows?
What?
Who knows?
Yeah. But so Oscar, do you have a set of cleaning scripts comparable to this?
Well, I don't know. I haven't found myself in such a big problem.
Bumblebees are
you sh-
probably nicer.
Yeah, so that's that's sometimes that's easier when you don't need that much data. Or on other cases, other people are doing the work, so you just did data. But yeah, I do have a small script that is started just because I wanted to download some species from GBIF, but it was, I don't know, per species 100 or 200 records. So by the end of the day, I had like a 50,000 database, so I didn't find that hard. And I was not using, for example, my filters are like only from a collection, right? So I don't use any photos or stuff like that. So that makes my data less and less big. So that's why I haven't found so much of that data, like James was talking about, right? So yeah, that's why my script is kind of more simple, but I get it and I like it. I bet when I find myself working with 5 million records, then I'm gonna be like, "Hi, James, do you remember me?" Yeah.
Well, I'm hoping that'll actually be in a couple of months.
Yeah. This is a very specific question, but like, are you using like mostly dplyr, or do you also use data table? Just because it, like, for the number of records that you seem to be working with, I'm surprised. I don't know. Dplyr seems to be slower.
I haven't used data table.
Okay.
No.
I haven't either.
It's so much better for working with like this number of records. Like once you get over like, I don't know, hundreds of thousands or like a million, it does all of the same operations but way faster. The syntax is a little bit different, but yeah,
that's super cool. Yeah, I've mostly like been moving away from base as well. So I'm like, yeah, dplyr is faster, and this is great. I'll just continue that trajectory.
Yeah, I mean, I've dabbled with the same types of things. Eventually, like for the occupancy models, I'll end up using the data that Paige and Lindsay and you guys all cleaned. But yeah, using data table just makes all of these operations a lot seamless. Like just because I know you kind of briefly mentioned that the data is almost getting a bit too big for R, and that data table makes it so that it's not the case.
Yeah, that sounds good. It's not so terrible on my computer. I think the longest thing that I'll run in terms of the 12 million data set is like 10 to 20 minutes. But I've also got a pretty good computer. So I'm very aware that not everyone has 64 gig of RAM and like good cores. So I'm a little worried if someone runs it on their home laptop that it might shit itself. Excuse my Australian.
Um,
It's fun.
so actually, so Melissa, I've never heard of data tables. So did you say you use those as opposed to like just CSVs?
No. So it's a package. So like
Oh, okay.
you still have the data as a CSV, but
Okay.
the function to read data like fread from data table is a lot more flexible than like the read CSV categories.
Okay.
So if there's like a lot more mistakes and stuff like that, actually makes it easier. And then the filtering, the subsetting, the modifying in place, all of these things happen within seconds, not within minutes for these types of data sets. And so it just makes handling the data a lot easier.
Is it data.table, the package?
Yes.
I've got it installed, but
you should use it.
I don't know why I do.
But anyway, so what I'm really hoping is that once we, in the next couple of weeks, submit our US paper, that at the same time, Oscar and Angela will have made progress on getting a sense of ellipsoid versus maxent, and that we can, by May or June, start over again and like take our range models and basically make 4,000 plus species distribution models.
Yeah.
But in that process, we're going to want to go back. Our data is already almost a year old. We'll want to go back and get that data and other data and go through all this process. And so I would think that utilizing your process could make that a lot easier.
Yeah.
Okay. So, sorry, you had something, Paige? Or?
Oh, I just said I would agree.
We can use the USC
Okay.
clusters.
Sure. And I just assumed that. So, Oscar, what are your resources like for running stuff? Your computer resources?
Yeah. So far, I haven't encountered any problem with the computer that I have plus the computer that is in the unit. It's not that big. It's just like, because it was Max and it doesn't take that much to query stuff like that. So yeah, I have like i7 64 RAM, and it's not that big of a deal to run models. And I think we started like working with Angela on this ellipsoids, and her computer also handles it pretty well actually. So yeah, I was very impressed about that. Like she was like, yeah, in like 8 hours we already have like, I don't know, 100 species ellipsoid model. And I was like, oh, okay, cool.
That's pretty good.
Yeah,
it's not a problem then.
Yeah, that's nice. So that was good. And in any case, I guess the only thing that sometimes it's kind of hard to work around is when you're using like parallel or stuff like that, and you don't have a computer, right?
Mm-hmm.
That's basically it. But I don't think we're going to use that unless we choose another package. So yeah, pretty much it's really good. Yeah, when we get this like you got, like, it's the script that Angela has is really cool, like really nice to do ellipsoids. It's just like if you already have your data clean, it's just like, yeah, just put the computer to work and just go out and get some margaritas or whatever. It's really
So I wonder, it might be good to try to have a specific meeting, James, with you and get you and Angela together to see that, because so that's a good point, because my guess is Angela's probably using whatever Jorge and the Kansas group have developed over the years, and it probably is pretty sophisticated.
In terms of SDMs?
In terms of cleaning data, it's like they do so many SDMs. It's like, I can't imagine that they don't.
And I don't like just bad mouthing people willy-nilly